Cool, man. Hey, thanks so much for inviting me to talk. It's so fun. I really appreciate it.
Yeah, yeah, absolutely. It sounds like you're doing some very interesting work. I'd love to hear about it and then sort of brainstorm on it with you.
Absolutely. Um, so I guess just some background is to say that I've been I've been studying ants for pretty much my whole career, um, and mostly from the perspective of like molecular genetics. So my undergrad advisor pictured there, he found these genetic elements that can switch fire ant colonies from having a single queen to a few hundred queens in them. And this big story in the um, late 80s and early 90s, like the first example of the genetic regulation of social behavior, right? Um, and I've been kind of continuing on that thread, trying to identify natural genetic polymorphisms and then use them to understand basic biological questions, right? So it's almost a little bit like I think of it almost like a natural mutagenesis screen, you know? So the same way that like you have a screen where if you see a certain phenotype, you know that you've hit the pathway you're interested in. It's kind of like we're trying to do that with wild populations. Like we look for certain kinds of natural phenotypes that we think that phenotype has to result from the modification to some underlying process. Does that make sense?
Yeah, can I ask a question about the phenotype that you just described with 100 queens? Is it
Yeah.
is it a modification of ant behavior that allows more queens to develop, or is it actually pushing specific animals to become queens and the rest of the just sort of well puts up with it? Like where's the
It's um it's a profoundly beautiful discovery and it's very, very deep. It's one of these things, sorry, my application is popping. So um it's one of these things where it's really like a whole syndrome of features.
Hmm.
And it's more like any trait that you measure is like a fingerprint or an indicator of which state of the syndrome you are, as opposed to a description of the actual phenomenon, right? Um, and so the syndrome is basically that um, there are these two forms of colonies. One of them will have one queen in the colony, and one of them will have a few hundred queens. But the colonies with a few hundred queens, they also lack territoriality. So they don't form discrete colonies with discrete territories, but instead they form these kind of carpets of ants with colonies all next to each other. Um, and yeah, basically they generate a very large number of queens, but they also accept new queens back into their colony. And one of the big discoveries of this was confirmation of the green beard effect predicted by Hamilton and then later Richard Dawkins that um, you could have selfish genetic elements that act by perceiving themselves in other individuals and then killing or benefiting them. And this was the first demonstration of such a thing actually existing in nature. So what they found is that these queens that form these multiple queen colonies, they all have a genetic element that induces the trait. And then their worker daughters um, of some kind of altered behavior, some kind of altered pheromone perception, where if a queen has the supergene, she'll accept it, but if she lacks the supergene, she'll kill it. So all of the hundreds of queens in the colonies have to carry the
Hmm.
genetic element that induces it.
Wow. Wow.
Um,
Yeah.
and yeah, since then, I had this joke where we were studying ant genetics, you know, in the like early, um, late 2000s, early 2010s. And at the time, there were like 10 people in the world doing this, right? Ken being one of the first and me being trained under him. And so I had this joke that the people studying ant genetics had collectively discovered fewer Mendelian elements of any consequence than Gregor Mendel found by himself in his one pea plant in his one greenhouse, right? Um, and so it's really a new field to do just type of like molecular genetics with ants. It's not really, there's two reasons for it. One is there's technical challenges, but I think an equal important reason is just that it's not been the historical focus of interest to people studying ants, right? Um, but since then, now that we have like whole genome resequencing and stuff like this, um, people are mapping these genetic polymorphisms all over the place, and it's growing exponentially. So, you know, the second one, the one that Ken had was the only one for decades. The second one was found in, I think, 2012. By 2020, we had like six. This year, we'll probably have like 20, So we have tons and tons of genetic information now about different types of polymorphisms in ant species.
And how, just in terms of how many colonies do you need to look at to be able to find this? Because a lot of times in these screens, you would have to go through a lot of animals to actually see anything.
Yeah.
Is this just more prevalent, or are you guys better at detecting it, or like, well, how does that work?
Um, I would say that I would make the hesitant claim that most ant species lack the types of polymorphisms I'm referring to, right? That may be wrong. Maybe they mostly have them, but I think most of them appear to be monomorphic, right?
Mhm.
Um, but the key kind of observation is like a gestalt or an intuition that you go into a population and within a freely interbreeding population, you have like two discrete forms,
Mhm.
and you can even do like high school level Mendelian statistics where if it's a continuous trait, even if you have two forms, if it's a continuous variation in the thing that it determines them, you should have like a unimodal distribution. Whereas if it's a Mendelian trait, you'll have like a bimodal distribution with peaks on the Mendelian ratios and stuff like this. So you can get a vibe for like if it looks like a genetic polymorphism. And even without doing those stats, just by being in the field and looking at the ants, a lot of people can just get that intuition, right? But then for years and years, there was nothing you could do with that. And so it would be described in literature as like an interesting phenotypic polymorphism. But then now we know that like and it would be described as a polymorphism and assumed to be environmentally induced or induced by quantitative genetic variation. And then you would call it facultative, quote unquote. Does this make sense?
Yep. Yep. Yep.
So that was the prevailing assumption for decades. Um, Ken was the one who kind of questioned that assumption in his fire ants that was thought to be either polygenic or environmental and therefore facultative. He felt like, no, it seems kind of discrete to me. And then he did all these Alzheimer markers and found that one of them perfectly correlated with the trait, right? Before they even had microsatellites. Um, and but these days, all you do is you just find those two forms and you just sequence the whole genome and you look to see if there's a genetic element that consistently distinguishes the two.
Mhm. Mhm.
Does that make sense?
Yep. Yep. Mhm.
Um, so yeah, so this is the kind of the background. And a lot of these Mendelian elements, well, I think one thing worth knowing is that um, there's a double-edged sword to these things, which is that on the one hand, mapping them is incredible because you have a genetic element that's like necessary and sufficient for a phenotype of interest, right? But uh, so far, every one of them that's been mapped has turned out not to be like a SNP or a simple small genetic locus. They've all turned out to be what we call a social chromosome or a supergene. So they're very large, and you'll basically have a chromosome often with like large inversions and maybe a movement of a centromere and stuff like this. And the chromosome will basically have an X-like form that's like freely recombining and kind of normal, but then it'll have a Y-like form that's got like massively enriched transposons, suppressed recombination, and it's like all screwed up, and that'll be the one that induces the like alternative trait,
Mhm.
you know.
Yeah.
Um, and so it's almost like these discrete forms, like a single versus multiple queen form of these fire ants. It's almost like a sex in a
Mhm.
sense. Like it's like two alternative reproductive strategies, two alternative ways of being in the world, sort of, and it's not conferred by a single mutation, but it's a whole suite of things.
Mhm. And what just I'm sure it's a variety of things, but in general, what kinds of genes tend to be involved? I mean, what are these things? Are these things neurotransmitters? Are they hormones? Are they structural elements, ion channels? Like what kinds of things?
Well, we've never figured out the causality of any of them,
I see.
right? So they all have hundreds of genes in them, and the work that I've been doing is to take one of these supergenes and do this laboratory Evo Devo and really try to map the causal mutation or combination of mutations. But even for sex chromosomes, there were some key genes like the sex differentiation factor or the sex determination, like SRY or whatever, that were known. But the rest of the sex chromosome, even those, was a black box until recent like CRISPR methods and stuff in the last decade or so,
Mm-hmm.
right? So this is a hard problem even in model systems. Um, so yeah, so that's kind of like a background of this field that we're working in. It's very exciting. All kinds of crazy stuff is getting found every year. Um, and the the one that I've become particularly enamored with is it's actually a mutation that arose in a clonal species. So we have a species we keep in the lab called the clonal radar ant, and it's my PhD was based around making the first CRISPR lines of ants in order to show how ants perceive pheromones. To test that they use these odor receptors to perceive pheromones. And this is a clonal species, but unbeknownst to us at the time, its ancestor probably had one of these supergene polymorphisms in the population. And so that clonal genome um, it's a diploid female who just lays eggs that directly give rise to new diploid females,
Mm.
right? Um, and yet, in that diploid clonal genome, one of the chromosomes looks like a sex chromosome. It has these two highly distinct forms. Um, and we found a mutant that has a large loss of heterozygosity mutation along this chromosome. So I often draw it like this. Um, it's like this. You go from uh, a heteromorphic chromosome with two very different forms to a loss of heterozygosity, where now in an interval of the chromosome, you don't have the heterozygosity, but now you just have two copies of the one form.
Mm-hmm.
Does that make sense?
Yep. Yep. Yep.
And this mutant, it does something terribly interesting, which is that it switched it from a free living uh, organism to an obligate parasite of its own species. Um, so uh, what it does is normally the small bodied ants will develop into workers and the large bodied ants will develop into queens. Um, in this mutant, the small bodied larvae, they have the same size and developmental time and external characteristics are completely indistinguishable. But at the onset of metamorphosis, they have overgrowth of all their tissues and they differentiate into this like parasitic queen phenotype. Um, and they don't resemble normal morphological queens. Like we show that they're very different from queens of related species, but they aberrantly express queen-like traits in small-bodied larvae that would otherwise become workers.
Mm. Is it just thinking about the morphogenesis for a second? Are they going to a different like how different morphologically are they? Is it are they really quite different, or is it just a different path to the same queen-like morphology?
Let me show you. Can I show you some slides,
Please, yeah,
or even just figure?
totally, yeah.
So project completed. Here, can you see this?
Yeah.
Oh, this isn't what I wanted. Here. So here's our mutant. So that's the wild type of this species. And this is often the evolution of clonality in ants is accompanied with the loss of the morphological queen caste,
Mm,
because the queen is the primary reproductive of an ant colony.
mm,
And so if you can clone yourself, you can just have workers that directly give rise to new workers, and you don't have a need for a queen anymore. That makes sense?
mhm, mhm.
So we're working with the clade that ancestrally looked like this, which is typical for ants, as worker and a winged queen. This is what the species was like when we found it. And then this is the mutant that originated.
Mm,
And so that's the picture of them. And this gives you a kind of a picture of what it looks like. Here are two close relatives of this species, and I've kind of traced them in Illustrator and stretched and compressed them to make them aligned to each other. And so um, basically the queens are kind of discretely larger than the workers of these other two species. Whereas in our mutant, we have this aberrant queen-like mutant that's broadly overlapping in body size with the wild type that would resemble the worker of the other ants.
mm, mm.
And so that's what this is showing, that the workers are smaller than the queens of these other two species, whereas the mutants and the wild types are broadly overlapping. And then this is showing that if you do a principal components analysis with two principal components, the first one captures size, and then the second one brings in this caste information. And so it pulls the wild type of our species with the workers of these other ants, and it pulls the mutant of our species with the queens of these other ants.
Mhm, mhm.
Um, so what was I trying to get to? Yeah, so this phenotype is really amazing, and it's the first intraspecific polymorphism for what we call a workerless social parasite. Um, but these workerless social parasites are known to emerge directly from free living ant species um, all over the phylogeny of the ant. So I was talking to my colleague last week, and I think he said that he's got at least 83 independent evolutionary origins of this trait, which is a lot. Most of the independent origins actually only have one species associated with them. Some of them have radiated into clades with a handful of species, but they're evolutionarily young, kind of a dead end strategy. Um, and But in every case, except for this one, all of those origins are a species. It's a distinct evolutionary lineage with sexually producing organisms with uh, no interbreeding between them, right? But in this case, we're going in a single step within a nominal species from the one to the other. And we think that's because this supergene probably conferred this as an intraspecific polymorphism in the sexual ancestor of the organism we're studying. We can't prove that, but that's the most parsimonious explanation. So, so it's cool from an evolutionary perspective because it tells us that um, it gives a really nice picture about how these parasites evolve, because it's been a big mystery for years and years. Even uh, like Darwin and the earliest ant natural historian William Morton Wheeler, E.O. Wilson, they all extensively discuss the mystery of the evolution of these ant parasites, because it looks like because they're parasites obligately of their host, and their host is often their closest relative, and so it looks like they have to speciate, like they have to become a new species out from inside another species without ever having an isolated population and any obvious way of preventing gene flow between the two forms. But now we know about these supergenes, and so there's an easy explanation, which is that if the supergene confers these alternate phenotypes on a single chromosome, then there's no recombination of that genetic element that's inducing the alternative trait. And so you can actually get divergence of two forms inside a single population, and then you can attach a gene that confers reproductive isolation or something onto that social chromosome, a million years later if you want to, and speciate it, or you could retain it as an intraspecific polymorphism. Does that make sense?
Yep, yep, yep.
So that's kind of what this paper was. It's like a cool a new form of natural history variation and a plausible explanation for this mystery about where these parasites come from, right? Um, but uh, the real thing that I'm interested in about this mutant, and the reason why I reached out to you, is because what I think is amazing about it is that the whole body is basically differentiating as if it were a large-bodied individual, an individual who's actually a small-bodied individual, right? So it's this radical decoupling of the relationship between tissue growth and body size that occurs normally in ants. And we have some other research showing that um, this relationship between cast and size is extremely deeply conserved. You might even be able to show that there's a single allometric function that relates size and morphology that is invariant across all of the ants that explains the worker queen distinction, and that this mutation and the evolution of these other parasite species represents like a breaking or like a secondary loss of that relationship. So yeah, so basically what I've been trying to do is kind of one to figure out why the development of ant casts in general shows this pervasive relationship with body size, why larger individuals differentiate in a queen-like manner and smaller individuals differentiate in a worker-like manner, just in general across the ants, but also um, what's broken or what has changed in this mutant to cause these small individuals to differentiate in a queen-like way instead.
Do these guys do these species build any kind of a nest or a structure?
Hmm. Yeah, what are you thinking?
And what does that look like? So I'm interested in the relationship between morphology and behavior, obviously, but at multiple
Hmm.
scales, right? So there's cell behavior makes embryonic morphology. Yeah, animal behavior. I've been looking at some basically the behavior of making and upkeeping a nest structure in terms of, for example, regenerative response, right? So there's some scenarios where you damage a piece of it, they'll go and rebuild it, and
Hmm.
the mistakes that are sometimes made in rebuilding it are actually very similar to to morphogenetic errors that cells make when they rebuild a body. So that you'll see duplications, you'll see basically misperceptions of distance and things like that.
In nest building organisms you're referring to?
Yeah, yeah. There's a certain.
Yeah,
I mean, I don't. Okay, I'm not 100% sure it was ants as opposed to termites. It might have been termites,
yeah,
but that kind of thing. Yeah.
that's really pretty. So these ants aren't really like complex mound building ants. It's actually the clonal radar ant, as with most of the ant species that people study in detail, is an invasive species that's found all over the world from human activity. And these invasive species tend to favor disturbed habitats and have relatively simplistic nest architectures. I think there's like a secondary loss of some of the complexity that allows them to like grow and spread faster. Um, so the ancestors of it probably were doing something more sophisticated, but we don't have field studies of them. Um, but this one, um, it's forming little kind of clumps in the ground, you know. Um, but it's a cool point because I agree with you that well, there's physics papers about these fractal branching architectures and how an ant colony or whatever can be similar to um, circulatory system or whatever. You know what I mean?
Sure.
Um,
Yeah.
and so yeah, so basically, I think that I think the point is that we have this mutant that's doing this really crazy thing, right? And um, it's a detective story from the genetics to try to figure out uh, sort of the proximate cause. And there is an approximate cause. Like there is some gene that is differentially expressed that will be responsible for this, right? Or different in activity or a combination of them. But there is at some level a genetic event that flips that switch, right? Um, but that answering that kind of proximate question might not really be the satisfactory answer to this problem that we're looking for because um, at some deeper level, I think there's like some kind of system or combination of system that's coupling like tissue growth and body size, and that could involve intelligence or that could involve collective behavior, different things. I'm not any way dogmatic about what it is, but there's always this nagging question of like, even if we solve the genetics, we might not really solve the question that we're really interested in here.
Yeah.
Does that make sense? And
Absolutely.
um, how do you go looking when you have this network and all these connections and all the layers? Kind of how do you go looking for that part that is really necessary and sufficient for this, as opposed to something that's kind of hooked into it, and another way of saying it is even if we saw the thing in the middle, the magic thing, sometimes I call it the allometry machine. Like one of the things we discuss in lab meetings sometimes is like, well, what would an allometry machine even look like?
Mhm.
Like how would we even, if we found one, would we know that we even found it?
Yeah.
You know?
Yeah.
And so that's the kind of stuff I wanted to discuss with you because I felt like the way that you're thinking about this, and I emailed you after watching one of your videos online about like embodied cognition and this kind of hierarchical structures of intelligence, and
Yeah.
um, it's very much like what I think these ants are doing, not from a aesthetic or personal view, but empirically, I have various reasons to think that some kind of architecture like that is required to explain what we're seeing. Um, and But then the question is, how do you get from that to the empiricism? Like what are the predictions you make, and how do you actually try to pull this thing apart?
Yeah. Um, yeah, there's a few interesting things. One thing, let me see if I can I'm gonna share this. You've probably seen this. Have you seen these things before? These are Darcy
Of course,
Thompson's.
yeah,
So like that's kind of interesting to me. This idea that what you're doing to get across these different species is deforming a grid according to mathematical rules, and this question of so what the heck is the grid, right? In real terms, trying to map this onto developmental mechanisms. I mean, nobody still has anything like that. Um, but I think that this issue of what should the machine look like is people, a lot of people assume that it's going to be the answer is going to be some kind of set of genes, right? It's going to be right,
yeah,
some kind of a genetic structure. You can go past that, and like we've been recently publishing some things on the genome as a kind of generative model, and specifically the idea that the genome is basically a set of prompts that are interpreted by the layer that sits between the genetics and the phenotype and the behavior. So that developmental layer, seeing it as a very active problem solving uh,
for
computational
sure.
layer, right? Not a a straight mapping from and it's and specifically the idea being that it's not just the mapping between the genotype and the phenotype isn't just complexity. It isn't just emergent, sort of feed forward emergence, but it's that middle layer is literally intelligent in the sense that it navigates a problem space with certain competencies to solve specific problems, and therefore the interpretation of the genome is not fixed. It's from the perspective of an observer, and that in that case being the developmental machinery. So there's that, and you could go even further and say that I think many things in biology, the actual explanation is not to be found in the physical space. It's found in some kind of mathematical space. In other words, right? So the actual answer to why is this like that is going to be often the answer is going to be a piece of mathematics. It's going to be because that's how prime numbers are distributed, or because that's because NAND is a specific kind of universal logic gate, or because Feigenbaum's constant is what it is. It's going to be
Yeah.
these things that are they're just not the kinds of things over which genetics usually spans. So,
Or another way of putting it is that the genetics is constrained to working with certain kind of properties of how systems, operate, right? Like,
yeah.
um,
I mean, so yes, and a lot of people do think of it that way as constraints, but I tend, I mean, and that's true. Of course, it's constrained by it, but I sort of flip it in that I think better than constraints, it's actually you get a lot of free lunches that way. So because a lot of these facts of mathematics, of computation, and so on, they give you things that you otherwise would have had to do a lot of hard work evolving. They give you for free certain properties that otherwise you would have to sort of micromanage and get for yourself. And so from that perspective, I think that what evolution is doing is taking advantage of these things. Yes, it's constrained by them, but it's not just. I think it doesn't do it justice to think of it as just constraints. I think it's actually an incredibly rich pool of resources. When
yeah,
you
it's like a river it's following. I'm with you, dude. I get it. I mean, the example of this that I often think about is that like when I learned about like Hox genes in the like early 2000s, it was like this idea, and maybe this isn't what the people who were studying it thought, but at least learning about it, this is what I was taught, is that it's like a program. It's inducing a program to make an eye or a wing. And oh, if you induce the wing program in the eye primordium, then you can get a wing to try to grow out of an eye or a leg or whatever. But I think that, my view on it now is that it's more like it's just a place indicator. It's just telling you you're in the tissue who makes a wing. And then knowing that you're in the tissue who makes a wing, there are lots of ways to respond to that information to build a wing, and it's going to differ from different cells. But it's not really telling it what to do. It's more just telling it where it is and therefore giving it a goal of what to do,
yeah
you know?
yeah
And I think that ant colonies, pheromones in ant colonies were again thought to be like verbs, like telling you to do something. Um, and there's a few, even those maybe not. There's a few examples of that, like a sex pheromone or an alarm pheromone. But the overwhelming majority of pheromones in ant colonies are really labels. Like they know who's the queen, who's the worker. Within the worker, they know if she's a nurse or a scout or a forager. They know all the developmental stages of the larvae, what their sex is, what their age is, and what their future caste is going to be. And I think it's mostly just a way of um, labeling everything, and so that the individual worker can decide how to engage with it more than it is telling them exactly what to do. You know. Um,
mm-hmm.
so I mean, another way of putting it, I always think is funny is like, a job, like the job description is not very important, but the job title is pretty important. Like
yeah
it's important to tell you, well, what's your job title? And then you do what you think you need to do to fill out that role. But the description of the detailed what you should actually be doing on a moment-to-moment basis is hard to really to predict or know. You
yeah
know,
for
um,
sure, and we see in morphogenesis, we see all of this. We see creative problem solving where you have a goal and the system, under different novel scenarios and perturbations, it will reach that goal using different molecular mechanisms if it has to,
yeah.
right? So it actually dips in to the affordances, the genetically provided affordances that it has, and picks what it needs to solve the problem. We have other examples like the Hawks thing that you were just talking about. So uh, if you induce an eye in the somewhere else in a tadpole, which by the way can't be done with the master eye gene PAC6, it only works in the anterior inner rectoderm, but with a particular voltage state, you can actually do that anywhere. But what happens is that it's a very simple signal. It doesn't remotely say how to build an eye. It's a very simple trigger. It's just, this is where the eye goes. And then there's this conversation between the cells that we've injected and the surrounding tissue, because we could inject very few cells. There's not enough of them to make an eye. And so what they do is they talk to their neighbors and they try to recruit them. And sometimes when you take a section through these eyes, you see that it's only about 10% of the cells are the ones that we injected. Everything else was recruited by them to do this. But at the same time, all the surrounding cells are telling these cells, no, you're wrong. You should be making skin or muscle or whatever it's going to be, because that's basically a cancer suppression mechanism. Is like for the neighbor, if you come across an aberrantly voltage to cell, you try to get it to normalize. So they have this they have this sort of debate back and forth about what are they going to do, and eventually it settles down one way or the other, and either the whole eye completely that eye field that we've established completely disappears, or conversely it wins and it recruits a bunch of its neighbors and it makes a nice eye that's only partially modified by us. So there's this like active interpretation and attempting to, I mean, it's kind of like what's central to being a collective intelligence is that you've got parts that are aligned to the same story, and that's in these processes trying to figure out what is the decision making at the level of the subunits, and then collectively what are they aligned towards, and how do they become aligned to specific outcomes. And it's like in development, it's you see this at the very beginning, because when we say there's one embryo, what are we actually counting? What is there one of? I mean, there's hundreds of thousands of cells at some point, and what you're counting when you say there's one embryo is that they're all aligned to the same morphogenetic goal that they're going to try to reach, even though you're cutting it and pasting it and introducing all kinds of weird stuff. They're all aligned. That's what you're counting.
Yeah. So you're speaking my language. I mean, I don't have any issues with that, and uh, the idea of the one cell needing to recruit the others, and either they do or they don't. Um, that I think there's something like that happening during allometric scaling, during development as well, where like you can have a developing fly and you damage the wing disc,
Mm.
and then it sends out this modified insulin-like peptide that can basically arrest the growth of the rest of the animal and give the wing disc time to catch up,
Oh, that's awesome.
right?
I love that.
Um,
I didn't actually know that example. Could you when we break, could you email that to me?
yeah, it's a beautiful finding.
That's very nice.
And uh, this is a little bit abstract, but the the organ body scaling is like a spectrum, right? So you go from small body size with small tissues to large body size with large tissues, right? And it's really interesting to me that it's a one-dimensional spectrum, kind of like across all animals. Like you take a principal components analysis of individuals of the same sex and genotype, and PC1 always explains like a lot of the data, like 80 or 90% or more, which is weird because there's a lot of tissues, um, and body size is just one variable. So you could have an n-dimensional space. It does not have to be largely one-dimensional, right? Um, and the way that I think about it is like almost like a harmony where like, you know, imagine that you're at a certain body size and you have a wing that's like a little bit bigger than it should be, or let's say smaller, like it's damaged, you know. Um, so the wing's a little bit smaller than it should be for that body size. It kind of has dissonance with the rest of the system and says, I'm off by this delta. Um, right. And then it doesn't just fix itself. The whole system also is like, oh crap, you're off by that delta. So maybe everything moves down a little bit while that one tissue moves up a lot. You know what I mean? And then everything is back into balance now. It's like they're all trying to kind of keep on some kind of,
Yeah.
the point is, and but I'm trying to figure out, and again, I think that this stuff is testable. It's an empirical question, but and I've got this mutant, but I'm trying to figure out what is the right way to approach this, because like, you're talking from the starting point of having this electrical signal that seems to be doing this stuff. And who knows, maybe you'll say, oh, I think I need to check electrical stuff in this context, right? But there's an arbitrary number of things I could check, and so it's hard to know, yeah, that's the kind of thing I wanted to discuss with you.
I see. Well, I mean, the reason I would say to check electrical is not. I mean, bioelectricity isn't magic on its own, but what makes it really interesting and important is that it's a very convenient cognitive glue. It's a really convenient way of binding subunits towards common purpose. And obviously, evolution caught on to this as far as the brain and nervous systems, and we caught on to this for computer architectures and so on. It's an incredibly convenient piece of biophysics for doing the job of a lot of scaling individual subunits into networks that can store goals, preferences, memories, decision making algorithms better than the individuals can. There's many other ways. I'm sure there's other ways to do it. I mean, of course, biochemical and biomechanical signals can do some of that. Maybe they can do all of it for all we know, but bioelectricity is a very convenient way to do that. So when you're looking for mechanisms that underlie problem solving, collective intelligence, some kind of reprogrammability, novelty, and all that, bioelectrics is a good place to look for that reason. And so, this is one of the things when I was asking about the genes is like, I'd be interested in these supergenes. Like how many of these things are ion channels, for example?
Our mutant, we've you know, it's been this 10-year detective hunt, right? So first we first found this weird chromosome and mapped the mutation to it. Then in development, we figured out that the early development is identical between the genotypes and that the genotypes start to diverge basically at the onset of metamorphosis, right when the tissue proliferation is happening. Um, and then at that time point, we find that there's all these genes involved in steroid hormone signaling that are differentially expressed. Um, and it's a really interesting finding because um, they're genes that are known to be differentially expressed in the context of um, the key steroid, in fact, the only known steroid hormone of insects, which is called ecdysone, basically the insect version of estrogen. Um, but the if you agonize the receptor in other systems, or you look at the upstream targets of the receptor, none of those genes are differentially expressed. So even though we're associating with the pathway of this hormone, it's not via the canonical receptor that this hormone signals through, right? Um, and so we had this good link that it's probably the hormone. I spent, a year or two thinking it would be the receptor, and then it ruled that out. And then we find that um, the most differentially expressed genes at our relevant time point inside the supergene are cytochrome P450 enzymes that are only expressed in the synthesis gland of ecdysone, the steroid hormone synthesis gland called the prothoracic gland.
Mm.
And so I've got this vague model now that where that the normal function of this hormone would be to signal via its canonical receptor to regulate developmental timing and body size. But I think that it might be signaling via some non-canonical receptor to regulate tissue growth. And if you build this type of feedback loop like I have here, you can explain a bunch of different stuff that we've observed, basically. So I do think that I haven't seen anything bioelectrical looking. But of course, it gets to this idea that the proximate genetic cause that we're finding could be hooking into some larger system, right? Um, uh, and again, like with your frog example, it's like, yeah, sure, maybe your mutant ox gene knockout or hox gene substitution, that there's just no genetic route to getting that electrical perturbation that you got. It might be, you know what I'm saying?
Yeah. Yeah, I mean the two-headed planaria line is an example like that, right? So in planaria, there is no stable mutant. There are no stable mutant lines for planaria at all. So unlike Drosophila and and every other model system, and I think we finally understand why that is. But it's an amazing thing that there is no aberrant line of planaria except for two lines, and we made them, and they're not genetic at all. There's nothing wrong with their genomes. There's nothing different about them.
Mhm.
And one has two heads, and one is confused about how many heads it's supposed to have and is random one or two. And in both of those cases, like you can imagine, if we had put them in the river somewhere, and 100 years later some scientists came along and scooped them up, they'd say, oh cool, where let's sequence the genomes and see where the speciation event went.
Yeah.
And that's not where it is, right?
Mhm.
So I'm sure, and I'm sure that's just that's the one, that the example that we've worked up, but there's got to be many others. So it could be things like that. I'm
And
curious.
and and let me just say that part of the reason why I think that our queen-like mutant is so interesting um, is because uh, I can't find a mutant in any model organism that produces this phenotype. So, like a small-bodied fly, there's many mutants that can make a small-bodied fly, but I'm not aware of any where it's a small-bodied fly, but all the tissues grow as if it were a large-bodied fly,
Mm,
right?
mm,
Oh, and in human genetic disorders, like they're genetic disorders where like you'll have like overgrowth of a certain limb, but like a systemic thing where like your body size is small, but all of your organs grow as if you're big. I don't think that there are any human genetic disorders that look like that, or
mm,
mouse mutants, or even like a C. elegans. I don't know how you'd know, but
mm,
and I think maybe people have seen mutants like this and they just didn't analyze it through this way of thinking. And so maybe there are mutants like this, but that's why I think that this is so interesting, is because this coupling of size and phenotype is a pervasive thing across the tree of life.
mm,
And when you find a mutant that breaks a phenomenon of interest, that's how you find a circadian clock, or that's how you find like, these fundamental things. And I think that um, this could be something like that. Like this could be hitting something that's really conserved and deeply found across biology, but just for whatever reason, it's not easy to find it with like mutagenesis screens or the traditional approaches people are using, you
yeah,
know?
yeah, yeah,
Um,
totally.
yeah.
What is the just out of curiosity, when you make ants of different shapes, what do the other ants make of this? Like, to what extent is it all based on chemical? Is it visual? Is it how,
Yeah.
do they know when something's a weird shape, and when do they care?
Um, so this isn't a direct answer to your question, but related. There's a common phenomenon in ant colonies that's called reproductive policing. So,
Hmm.
for example, a lot of species will only tolerate a single queen in the colony.
Hmm.
And if another queen tries to start like mate and start laying eggs, like maybe one of her daughters, the workers can smell, and there's a whole decades of chemical ecology work showing that when you become reproductively active, it changes your hydrocarbon profile on the surface of the cuticle, and then that's perceived as a reproductive pheromone that, in the context of the good queen, it coordinates behavior and so forth. But in the context of one of these extra queens, it'll lead to it getting killed. You
Mhm.
know, and yeah, like it's a you want me to be like really trippy for a second? It's like this like feminist idea that like gender is the social meaning of sex, that like to some extent uh your gender in society is dictated by how society interprets your sex, how you're treated, you know and and so we have these morphological casts of ants that also have distinct behavioral profiles, right? And there's the question about, to what extent is their distinct behavioral profile programmed by their morphology, and to what extent is their behavioral profile like the social meaning of their morphology, right? Um,
Yeah.
and I mean, so it could be that even if you're like a worker ant or a soldier ant, maybe some component of your behavior is kind of dictated by your morphology, but maybe some component of your more behavior is dictated by the way that other ants in the colony treat you,
Yeah.
you
Yeah.
know?
Yeah. Yeah.
Um,
I mean, that's the kind of thing I'm getting at, is some sort of a morphogenetic or behavioral chimera where, like, if we do introduce strange new phenotypes, let's say you bioengineer an ant that's just like completely different, uh what are going to be responses? I'm like other papers before, but we're working on this particular very kind of large review on collective intelligences and hacking of systems to try to infiltrate them, and mostly from the like biomedical perspective, of course. But I think there's a lot of interesting these kind of model systems, as far as how do they detect invaders, how do they know what's normal, and what are the outcomes that happen when you do have a mix of different things that normally don't go together as far as evolutionary selection. So, like, we've done another review on chimeric organisms, that what happens when you put two different sets of parts together that have all made assumptions or not about what their neighbors should be like, you know.
mhm.
And if you were to, I mean, so biologically, you can imagine altering these things to have different shapes, but also technologically, like if you made, I don't know, artificial robotic ants that did specific things, would they be ejected? Would there be some kind of new emergent mode of cohabitation? Um,
Yeah. I mean, um, one example of this is that there's like a thriving community of ant keepers and, you know, ant farms and hobbyists who do like really sophisticated like rearing and stuff like that. Some of these people have even done things like figure out how to get ants to mate in captivity with weird combinations of temperature and UV light inspired by studies of reptiles that like no biologist has found or published, but they can like get them to do mating and captivity and stuff like this. Some of
Amazing.
these people are really cool, you know. And one of the things that a lot of people will do is they'll kind of coerce ants into forming like chimeric colonies of two different species. And so there's these like videos on YouTube and stuff of getting like two very unrelated species that would never
Mm.
mix under normal conditions to live together, you know. And to my knowledge, this hasn't been studied scientifically as to like what the behavioral consequences of this are and stuff. But you might imagine that, you know, like your chimeric mixture of cells, that maybe it goes one way or the other, you know, something like that, you know, that, but who knows, right? Um, One thing I would say is that, you know, most ants in a colony are inactive most of the time, right? And it's not like the mental model where like every ant is running around and perceiving stimulus and doing stuff continuously is kind of how people would think about it, but it's not really what it looks like. There's a few ants that are doing stuff, and the rest of them are just kind of sitting there, you know? So observing these dynamics can be subtle because you need to take a long view and you need to look at places where there actually are like variable dynamic behaviors that you could quantify and stuff like this. Not to say that you can't do it, but just that it isn't, it might not jump off the page, you know? Um,
Yeah, interesting. Hmm. I think there should be a lot of yeah, I wish I knew this stuff better. I think there should be a lot of parallels here with the kinds of things that we see in morphogenesis. Um, yeah.
yeah, and so I guess I guess one thing I'm hearing from you is that there's an interest in finding like you said, bioelectrical signals provide a convenient glue to bind subunits to a common purpose, right? So uh, looking for um, the signal that is being used to act upon, and that might be one of your recommendations for me with this allometric scaling problem, right?
Yeah, I mean,
Um,
there's a couple of things that I could suggest in practical terms. One is keep an eye out in all the genetics and all the sequencing and everything else that you guys do. Keep an eye for anything that's an ion channel, ion pump, or an innexin, which is a gap junction component in vertebrates. So just keep it. If you see any of those, definitely let me know, and I'll help you make some sense of it. That's one thing. Another thing that you might do, are you able to drug these guys? Can you put stuff in the water, like
mhm.
compounds? Yeah.
So
You
like
could.
we have our cytochrome enzyme that have a good candidate. We've tried titrations of probably like 5 or 6 different agonists and antagonists of
Yeah.
cytochrome signaling, and none of them, they just were like lethal, and we didn't get anything cool out of it.
I can
Maybe it would
suggest.
work eventually, but
Yeah, we should let's talk offline about different options. And I don't know how onerous these experiments are, so maybe you can't try too many things. But I could suggest some cognitive modulators, some bioelectric and some not, that you could put in the water to kind of. I mean, I'm really interested in communicating with the collective intelligence of the colony. We had a project a while back to try to communicate not with the individual ants, but with the colony itself. And what we wanted to do was train it, and I can kind of describe how that was going to be. Not the ants. We weren't going to train the individual ants. We wanted to train the colony. And
yeah,
I think from that perspective, doing the kinds of things to the collective intelligence that you would do in neuroscience to probe the right
yeah,
the neural collective. Yeah. So there's some good tools that you can use. I think that would be interesting. I can suggest some specific reagents. Hmm.
yeah. And then so that's one thing I kind of gleaned from what you were saying, and then it seems to me that the other one is um, this idea of introducing kind of gross perturbations and seeing like what the state that the system returns to would be. And
Yeah.
it seems like that's one of your other major inductive tools that you advocate.
Yeah. So there's the problem solving, and then there's the creativity. So the problem solving is we hypothesize a goal, we put barriers between the system and its goal, and we see how good it is in reaching that goal despite all the weird things you're going to do to it. So that's the kind of stable decision making and problem solving. But there's the flip side of that, which is I put you in a situation that you've never seen before, and now the question is, can you pick a new goal and implement something completely different? And we've seen this, that would be like our Xenobot or our Anthrobot kinds of systems where you do something to this. The genetics are still in our wild type. You haven't touched the genes at all, but you put the cells in a novel environment, and they will create some kind of autonomous, coherent living being that has structure, behavior, and in fact, now we've studied the transcriptomes and all
Yeah.
of that, right? That have never been under selection per se, because this thing has never existed before. There's never been pressure to be a good Xenobot or anything like that. And so the question is, right, how good are you at meeting the goals you already had that have an evolutionary history, and then how good are you at improvising on the fly novel ways of being that are not that at all, and that are totally new. So I love these kinds of ant models for the possibility to really probe that. And chimerism to me is, I don't know if you've seen this review that we had with Vasily Nanos, who's my grad student, but basically looking at chimerism as a tool to probe the nature of the collective intelligence. So you introduce components that have a different they come from a system with different goals, or they have different properties, and then you look to see what does in the cases where they don't get immediately rejected, of course, what are the properties of the collective, now of the chimeric collective.
Yeah. Well, so I know that we're short on time, but I think that the thing that I'm really curious about is uh, in the evolution, and you had this paper about um, how this type of intelligent processes and stuff shape evolutionary outcomes. And um, so I think that the question for me is kind of, if you have an evolutionary question, or you have like a deeply conserved morphological or evolutionary pattern, like this allometric scaling pattern in the ant casts, how do you use, if that's the question you're trying to solve, how do you use these types of tools and approaches to address that question? You know what
Mhm,
I mean?
mhm. Yeah,
Um,
yeah. Hmm. Interesting. All right. Um, yeah. Do me a favor too, and send some links to some of the key papers as well.
yeah, I will.
Yeah, and I'll put it up. I mean, I'll show it to my group. We have a good machine learning group that's now looking at a lot of these basic questions and collecting
Yeah,
intelligence and how to uh for the kind of basically the invariance between learning and evolution, right?
yeah.
And I think there might be some useful stuff. So I'll show it to them. I'll also put the links up with the video so that other people can see. Yeah.
Absolutely. And yeah, if there's anyone in your group that's interested in talking, like I live pretty close by, and I'd love to like come out or something, and just to see, because I'm actually I've been at Harvard for 5 years, and
Mm.
I'm starting a lab at the University of Georgia
Yeah.
next year.
Yeah. Yeah.
Um, and so I'm taking a bit of a pause getting stuff published and stuff, but really trying to
Yeah.
get set up in a new way, and really,
Yeah.
you know, rebuild in an intelligent way that moves forward and builds on what we have now. So it's a great time to receive input and design experiments and make plans, because I'm not nearly as constrained as I would be if I was in the middle.
Sure. Sure. Yeah. Totally. No, that's great. Yeah. And do you know Will Radcliffe?
I
So
do. I can't remember why. I know the name though.
yeah, so
Um,
I don't know if it's I mean, he's in Georgia too. I don't know how close physically he'll be, but he's another really good person to talk to. He's a brilliant dude, and he's got a lot of good work on evolution of multicellularity and things like that, which
okay.
I think ultimately are gonna impact on what you're doing. So yeah, all right, yeah, very
Hey
very.
man, I really appreciate it. I learned a lot, and this is
Me
really
too.
great. Thank you so much for
Yeah.
taking the time.
Yeah, thank you so much. Yeah, no, I learned a lot too. It's a really interesting system to think about. So I'll keep thinking,
Yeah.
and I'll show this to a bunch of my lab people. So thank you.
Sounds good, dude. Rock on.
Very good.